peptide bound to a class 2 molecule, then there's a CD4 T cell out there that probably recognizes that peptide in context of that class 2 molecule. And so the macrophage can find that T cell. The T cell was activated in lymph node, it circulates to the site of infection. Now the bidirectional interaction taking place there is the macrophage is presenting antigen to the T cell. The T cell is producing cytokines like gamma interferon that activate the macrophage into that super Pac-Man, super aggressive macrophage and can kill much better. Multiple applications, we won't be able to really get into those things much, but what you're going to see is, I'm not talking about clonal expansion a little bit later, and if you think about all the different white blood cells that are in your body, very few of them are going to be specific for a particular antigen. I'm going to give you a number 1 in a million, and that's just a number I'm putting out there, but that's not unrealistic. 1 in a million of the white blood cells might be specific for an antigen that you come in contact with. How do you get that 1 in a million to interact with that antigen? You have to work through this lymphatic system, the lymph nodes, the spleen, and those types of things. All right, so lymphocytes we've already talked about. You've actually seen this slide before. B cells get activated in the lymph nodes or spleen, the lymphoid tissues, and they can differentiate into these cells called plasma cells that then produce antibodies. T cells get activated in the lymphoid system, usually lymph nodes, and they, once they become activated, there's some proliferation that takes place, so you have more of them, for one thing. And then those that leave the lymph node go find the site of infection, they go cruising. And I'm going to show you something that kind of demonstrates, well, how do you get an activated T cell to the right place versus a naive T cell to the right place. A naive T cell, when we use the term naive, that's a B or a T cell that has never come in contact with this antigen. And it almost blows people's mind when they start thinking about how does a B cell and T cell get made. It is a completely random process that takes place in the bone marrow and the thymus. But it's an efficient process that allows you to have millions of B cells with millions of different specificities, and the same thing for the T cell. Then you just have to get it to find its proper antigen. Natural killer cells, not going to go into that much more after this. Large granular lymphocytes have granules. It says non-specific recognition, but basically they recognize some self molecules. So some of what they do is they go around and they kill virus-infected host cells. And sometimes they actually can kill cancer cells, but that's a little more debatable. But they're really there to identify an altered self cell, an altered host cell. And sometimes when viruses get in, they alter some of the surface molecules that are being expressed on their surface, and the NK cell recognizes that and kills it by apoptosis again. You release these granules, and the granules can get in there and induce apoptosis, so we don't have a lot of information. But the key thing is kill the virus-infected cell before it can make a lot more virus that can even get out and infect the host cell. The dendritic cells I mentioned, very potent antigen-presenting cell, APC. And this is what they've got these long projections. Why do they have those long projections? Surface area. That's the main reason is they can have lots of class 1 molecules, lots of class 2 molecules on their surface, can interact with lots of T cells and B cells for that matter to present antigen to them so they'll be activated. Some of them, I'm not going to get into interdigitating unfortunately. All you guys need to know is dendritic cells are really good antigen presenting cells. They're the most important at activating these naive T cells in the lymph nodes. And we'll talk about how they bring antigen into the lymph nodes and activate the T cells later. Macrophages we already talked about, they phagocytize foreign particles. Opsonized. This might be one of those terms that you've not heard of before. Opsonins are things that coat particles like bacteria or could be viruses, fungi, these types of things. Opsonins, and sometimes the opsonin is just an antibody molecule. Sometimes it's these complement things that I told you about. Complement is a term we use for just over 30 proteins, different proteins, that go through a cascade of proteolytic cleavage, and they do lots of things. And I'll go into it very briefly, but it's a critical part of the innate immune response because it attracts, the byproducts attract the inflammatory cells. They opsonize to enhance phagocytosis by macrophages. And it also, if you go all the way through, you form a membrane attack complex, which is a pore. So the component molecules can form a hole, actually many holes on the surface of these pathogens and cause them to leak their innards and die. And then you get a little more inflammation and you can kill them a little bit better. But that's, opsonization is basically the coating of something to make it more digestible, more easily phagocytized. I think of it as gravy on your potatoes. It makes it a little more appetizing and you slide it in, or maybe chocolate syrup on your ice cream, whatever. Something that makes it a little more easy to digest or engulf and go from there. Produce factors that recruit or activate effector cells. I mentioned the chemokines, and then we also get those cytokines that are generated following a PAMP binding to a PRR. You've seen this slide before. It's the same thing that you just saw, except the diagram. So I'll bring it up again. We have an infectious agent that is binding to a specific receptor on the macrophage, inducing this engulfment, this process of phagocytosis. It is inside a vesicle inside there, so it's completely surrounded by this vesicle membrane, and those can fuse with other types of vesicles like lysosomes that contain these proteolytic things, and also sometimes they have these proton pumps, and they're highly acidic. And so that acid can help kill and degrade the proteins that are in there. So that's your phagocytosis site. The activation site, again, is a signal is sent to the nucleus, a gene gets transcribed, or genes, and in this case, inflammatory cytokines get made and released into the environment, and the closest effector cells around that have receptors for it become activated to get involved in the infection. Macrophages also play a role in antigen presentation, especially at the site of infection. Not in the lymph node as much, but at the site of infection. So a T cell gets activated in the lymph node. It's this activated effector T cell that goes to the site of infection. Before it does something, it needs to bump into its antigen again, and it bumps into its antigen by running into a macrophage that has MHC class 1 and 2 on its surface with the peptide. Cytokines get produced by the macrophage because of the PAMPs and the PRR interaction. The cytokines help stimulate the T cell some, co-stimulatory molecules. These things will all come up a little bit later. T cells need co-stimulatory molecules when they're naive. They don't need them when they're activated, but the macrophage does provide some co-stimulation. Multiple checkpoints to make sure we don't have immune responses out of control. Effector cells and cell-mediated immune response, this is where the T cell comes in there, gives them some gamma interferon and activates them and makes them work a little bit better. They also play a role in humoral responses because they have receptors. And I'm going to put this up here. Fc, big F, little c, this Y-shaped antibody molecule that we talked about with 2 heavy chains and 2 light chains. This is the antigen binding part. So this is antigen binding. And this is exactly the same antigen binding site because the amino acid sequence of these 2 big chains are the same. Amino acid sequences of the 2 light chains are the same. This part down here is called the FC. And it gets its name from fragment crystallizable because of early experiments. They're trying to understand the structure, and it turns out that part will crystallize following some kind of protease degradation. But we still call it FC because this is the effector part of the antibody molecule. This is just recognition. And this is pretty typical of lots of toxins and things that are out there. Part of the molecule is for recognition. Part of the molecule is involved in an effector function. This FC part here can be bound by FC receptors on macrophages and other cells. And so when it says receptors for antibody and complement opsonized particles, that's what's going on. You have a pathogen that is coated with this antibody. And now the FC part of that antibody bound to, here's this big bacteria here that's bound by this. Now this FC part here can bind to a receptor, an FC receptor on the macrophage and help in the engulfing. All again, it's just to help the innate response work a little bit better. The complement opsonizers work in the same way. There are complement receptors on the macrophage that when the complement is coated, it's the gravy around this bacteria. It binds to it and can be taken in. So you saw this before. You'll see it again. This is what's going on here is when we get to this, the far side of the panel, we produce some cytokines. You can get some leukocytes or some lymphocytes like T cells cutting in there, and they can interact with the macrophage. You can get some antibody coming in there. That antibody can bind to those bacterial cells and then bind to an FC receptor and facilitate engulfment and phagocytosis, or even complement is getting in there. So that the vasodilation is going to allow the complement proteins, the antibodies from the serum, and additional leukocytes to get in there and help with the infection. Granulocytes. They're called granulocytes because they have granules. And they get their names from the way the granules stain, and it doesn't really matter. Most of them are very inflammatory. The neutrophils being the ones we talk about the most, because in fact the neutrophils are the most abundant white blood cell in the blood. They get recruited by chemokines. Sometimes the byproducts of the complement cascade are very chemotactic and can draw in these neutrophils and these other granulocytes. They get activated by cytokines at the site of infection. Typically, a macrophage that's already at the site of infection can stimulate the neutrophil cell. But there are certain types of T cells that produce other cytokines like IL-17 that will stimulate the neutrophils to become activated, but also cause more neutrophils to be produced and drawn in. And they will typically release cytoplasmic granules, kill microbes. Neutrophils can phagocytose the foreign particles, but the eosinophils and basophils that are part of the granulocytes, we don't typically think of as phagocytic. So here they are. The one on the bottom left, mast cell, I've put on there, but it's really, remember, we don't know where it comes from. But the neutrophil up in the top left, it got its name because a neutral staining, a neutral pH stain would stain it. The eosinophils and the basophils had eosin was staining them, and basic stain was staining the basophils. This is how they got their names, and so they've stuck. But they do different things, and so the neutrophil is, like I said, going to be very abundant and go to be one of the first leukocytes at sites of infection and just cause a lot of mess. When you think about pus formation, a lot of pus formation is neutrophils getting in there, killing the bacteria that's in there, and also killing themselves in the process. We make lots of neutrophils, but they kill themselves. There's collateral damage, some of the host cells. So all that pus is basically dead host cells, dead bacteria, and dead neutrophils that have gotten killed during the process of clearing the organism. Eosinophils, and we'll talk about each one of these a little bit more. Basophils have some other roles, and then you see the mast cell there. You all know what mast cells are, what they're good for. They're found in the tissues. They are an innate effector cell, so they don't recognize specific antigens. But for some reason, I'm allergic to wheat dust. Yeah, Destiny, I'm a wheat farmer, and I'm allergic to wheat dust. Can you read it? And Bermuda grass, but I still mow my lawn. And somebody else is going to be allergic to this, somebody else is going to be allergic to this. Well, how is that possible? These FC receptors that we talked about, there's a certain subtype, certain isotype of antibody molecule that you may have heard of called IgE. We'll see that in the next lecture, what the different types are. IgE goes and binds to the mast cells and sensitizes the mast cells. And so I have antibodies, IgE antibodies, against wheat dust and Bermuda grass, and I have mast cells that are coated with that. So when I come in contact with those allergens, the mast cells degranulate, and cause the sneezing and the coughing and all that other stuff that goes along with it. Well, what's the purpose? Well, I mean, why would we do that? It turns out that it's a mechanism to get rid of parasites that we may have breathed in or something. All that coughing, you're coughing that stuff out, you're sneezing that stuff out when you have allergic reactions. But because, at least in places like the United States, the number of parasites that we come in contact with is much lower. Because of the better hygiene, these mast cells don't have anything better to do, so they're building up IgE that's specific for something that we don't want to respond to. So allergies are because we have a very clean environment. It's something that we just have to deal with. Most numerous is the neutrophils. They have a multi-lobe nucleus, and this is where they get their name, polymorphonuclear leukocytes, or polys, PMNs. And I don't know what the significance of it is, but that's what they look like when you stand there. And they have these granules in there. They're going to be the primary cell at the inflammatory site. They have phagocytic properties. They also can kill extracellularly. Eosinophils also have an Fc receptor for IgE, but they do something different. Whereas mast cells degranulate and release all these things that causes allergic reactions, what happens with the eosinophils is, if you think about some of the big helminth parasites, I don't know how much parasitology you all had, but some of these worms are way too big for our white blood cells to engulf and kill. You can't phagocytize them and kill them intracellular. They have to be killed extracellular. And so eosinophils get IgE on their surface, specific for a particular helminth parasite, and they go and they bind to it and they spill granules. And so you'll have multiple eosinophils maybe landing on this worm or this other helminth and dumping their granules to try to kill that parasite and get rid of it. You do find in cases of allergies, but that's what they're really there for, is to kill these others. And I won't go into anything of this. Is the hematopoiesis. This just kind of reminds you of where some of these things fit in here. This slide's a little bit different, and because it's a little bit newer, but you can see, as I was mentioning earlier when we had the question, you can see here that the immature dendritic cells is here, and it's kind of not, you don't know where it's supposed to fit, but it kind of fits in the lymphoid side and in the myeloid side, and we still don't know the mast cell where it comes from as far as the precursor. And we'll move on. I know I'm going through this fast, but you've seen some of this before. Again, here's our lymphoid tissues, primary, bone marrow, and thymus. Why? Because that's where the B cells and the T cells get educated and acquire their receptors. The secondary is where the real work goes on to activate them. Trafficking of lymphocytes, and this gets a little hairy because what we're talking about here is having certain markers up on the surface. And when I say marker, it's really important. So a CD thing, we were talking about having CD, there's like 269 CD molecules. And you've heard 3 of them already, maybe 4. But these different molecules go up on the surface and they recognize another type of molecule. Maybe you've heard of integrins that recognize adhesion molecules. So there's a lot of different adhesion molecules whose job is really to get cells to interact more tightly. And so in case of lymphocyte trafficking, we're going to have lymphocytes going into the lymph nodes. And we're talking about B cells and T cells. Pathogens are being introduced in some manner, and they're being picked up. Well, the first few hours, the antigen is probably getting to the lymph node in a soluble form. It's just draining the lymph node as an antigen and gets picked up by something in the lymph node, a dendritic cell. Not very concentrated. Not a lot of antigen getting to that lymph node, so not a whole lot of activation going on. Later, the dendritic cells at the site of infection are picking up a lot of that antigen and processing it and taking it to the lymph node. And so they're going to be much better at activating the T cells in the general lymph node. So lymphocytes enter the lymph nodes. Somehow antigen gets transported to the lymph nodes, and the best is going to be through a dendritic cell that was at the site of infection, picked up the antigen, and brought it to the lymph node. The antigen-specific lymphocyte runs into its antigen and becomes activated, and now it sounds like you have this one lymphocyte interacting with this one dendritic cell, and then it gets activated and it leaves. it's much more complicated than that. There's a proliferation that takes place. Activation causes that one T cell to become 2, 4, 8, expands. Some of them stay there and become the memory T cells that are part of the secondary response. Some of them go to the site of infection. So this is when it says activated lymphocyte enters blood and travels to the site of infection. Some of them do. Some of them, those effector T cells, go to where the action is. And here again, this is just showing you the lymph nodes, the architecture, so that the idea here is that blue area where the T cells are, that's where the high endothelial venules are going to open up. And that high endothelial venule is just a place where cells can move out of the blood and into the lymph node. The idea here is if things are coming from the blood like B cells and T cells, they're kind of dumping out in the same place, and so they're kind of bumping into each other. You bring dendritic cells in there, and they will migrate to that place and interact with the T cells. But the idea is they're kind of running into each other as they're coming through the lymph node. It's kind of compact in there, and hopefully they'll interact and stimulate a response. Okay, so the lymph nodes, I think it says here, they collect antigens from skin, GI tract, respiratory, just about everywhere. And spleens has a similar type of, just say, architecture in that it's trying to cause interaction between the B cells, the T cells, the dendritic cells. But of course, it's going to look different. It has a white pulp and it has the red pulp. But and we're not going to get into the details of what a spleen looks like. Just keep in mind that it's a secondary lymphoid tissue that collects antigens from the blood. And the same things that happen in the lymph nodes are going to take place in the spleen. Dendritic cells are going to bump into T cells. If they have the antigen the T cell recognizes, the T cell will activate it, it will proliferate, and it will differentiate into that memory group and the effector group. And they'll find where the antigen is. And this is just showing you a more detailed version of it. I don't think this one isn't showing high endothelial venue, but what you do see way up at the very top, you see a dendritic cell, and you also see a bacteria. This is kind of showing you that from the afferent lymphatic vessel coming into that lymph node, that's why it's draining. That's the direction the lymph is flowing. So this is the draining lymph node from the site. So the bacteria or the antigen from the site of infection is coming into the lymph node. The dendritic cell has already picked up some of this antigen. It's coming from the site of infection as well. And it's going to get dumped off right there in that blue area where the T cells are. And B cells are going to be coming through there as well, and they might run into it as well. But what ends up happening is you can see that dendritic cell in the middle there that has about 6 blue and more than that of those purples. What's happened is some T cells have recognized their antigen on that dendritic cell and have become activated and proliferate. Some of them leave, and you can see there they go. They're flying out to go in search of the antigen. Some of them are going to go through the lymph and that route. Some of them are going to get back into the blood and go in search of the infection. The other side, the left side, is showing you that B cell rich area. And so the B cells go through the T cell rich area and migrate out there, and they'll form a germinal center where they'll start proliferating if they become activated. And if you look, you can see some blue dots in there, blue cells. Those are the T helper cells that have moved over there and are trying to help the response a little bit more by providing certain cytokines. Differentiation that takes place is plasma cells, and this is showing you some plasma cells staying but some leaving, and they're going to go somewhere where they can produce lots of antibodies. So you'll get some antibody production in the lymph node, and it'll get into the blood or into the lymph, and you'll get some plasma cells going somewhere where they'll make lots of antibodies and get it circulating throughout the body. Because the antibodies don't know where to go. They're going to go wherever they're allowed to go. You make a whole bunch of them, and they're going to get into where the vascular endothelium is opened up. So the idea here, and this is where it gets a little tricky, you want to keep the naive T cells moving through the lymph nodes, sampling antigen, lymph nodes and spleen, while you want the effector or memory T cells going looking for the sites of infection. So naive T cells in the lymph nodes moving from one lymph node to the next. Memory or effector T cells to circulate through the body. Memory T cells not so much circulating, but they don't have to run into their antigen right away. The effector T cells are circulating throughout the body looking for their site of infection. And these are some ways that we kind of visualize this. We call them homing receptors on lymphocytes. And this is not as popular as it used to be, but we have homing receptors on lymphocytes, and we have addressins on the endothelial cells. And it's supposed to be like, okay, you stamp this particular T cell, and it has I'm supposed to home here to this particular address, and it goes there. And so that's kind of what we're thinking about right here. So the lymph node, high endothelial venule, that place where the T cells move out of the blood into the lymph node, has a particular addressing or zip code that is recognized by a particular molecule on naive T cells. And I'm going to show you those in just a second. Versus that inflamed vascular endothelium that I've shown you about 3 times, where that macrophages cause vasodilation a little bit, and there's swelling and there's redness and all this stuff. There are different zip codes or addressings on that vascular endothelium than there are on that high endothelial venule endothelial cells in the lymph node. And so the naive T cells recognize the ones in the lymph node. The activated T cells recognize the ones out in the periphery where the infection is taking place. And you keep the cells going to the right place. So naive T cell enters the lymph node, sees its antigen, proliferates, and becomes activated. The activation will downregulate some things and upregulate other things, but this is upregulates other adhesion molecules that you would find on effector T cells. Now they go through, they will recognize a different type of endothelial cell to attach to. The activation then causes the loss of those same molecules that were involved in getting it to bind to the high endothelial venule of the lymph node. And the activated T cell then leaves, the activated T cell now has the different adhesion properties, and it'll leave the lymph node and go into the blood. And I'm going to show you pictures of those right here. This is a naive T cell. You guys should have both these showing up, but the naive T cell has, and I don't expect you to remember sulfate, sialyl, Lewis X, glycoproteins. That's what the L-selectin is recognizing. I would expect you to know that naive T cells have L-selectin on their surface. The selectins, there are 3 of them: L-selectin, E-selectin, and P-selectin. And they're called selectins because they are lectin-like molecules. And you all know what a lectin is? Lectins bind carbohydrate moieties. And these are C-lectins, the calcium's involved in the binding, but L-selectin, L is for leukocyte for our purposes. L-selectin is on the naive T cell, and it recognizes certain carbohydrate moieties that are only found on molecules on the high endothelial venule of the lymph nodes. There's actually more than that, but this is what the naive T cells recognize, is this on the high endothelial venule, those endothelial cells where they cross into the lymph node. They're going to lose that once they become activated, because activated have something else. T cells, all T cells have LFA1 on their surface that recognizes ICAM1 on antigen-presenting cells. That's a stabilizing molecule. That interaction stabilizes the interaction between antigen-presenting cells and T cells. But on the bottom, you have an activated effector T cell. It no longer has L-selectin on it. It has something called VLA4. VLA4 recognizes VCAM1, vascular cell adhesion molecule 1. Where do you find VCAM1? It gets upregulated on those endothelial cells where that macrophage has interacted with the bacteria and produced cytokines. It's that inflamed vascular endothelium. So you have 2 changes going on here. Naive T cells come out of the thymus with the ability to bind into the lymph node. Once they become activated, they lose the ability to bind in the lymph node so well, and they gain the ability to bind to inflamed vascular endothelium. And so they leave the lymph node, they go out, and they happen to bump into this infection, this area of infection, and they bump into an inflamed vascular endothelium and combine. Yep.
Do you mind saying that one more time? So they lose lymph binding and gain.
You got it, where the inflamed vascular endothelium is. So they lose that L-selectin, which allowed them to get into the lymph node, but they gained this VLA4, which allows them to bind to an addressant that's found on inflamed vascular endothelium, sites of infection. And so they bind, and that's a specific interaction, VLA4 and VCAM1, but it's not antigen specific. It's just ligand receptor specific. And the ligand on the T cell is only going to come up in the activated state. The receptor that that ligand binds to is only going to come up on the endothelial cell in an inflamed state. And that's how you maintain some interaction between getting the right T cells to the right places. And this is just showing you again, naive T cells bind behind the L-selectin, the effector T cells bind to activated vascular endothelial sites of infection. And, high endothelial cell on the left, activated vascular endothelial cell on the right, and you can see the VLA4 VCAM1 interaction, L-selectin binding to something on that. I don't care that you know CD34 or sulfated sialyl-Lewis X carbohydrate, just L-selectins on the naive T cells helps them get into the sites of infection. Now you're going to see L-selectin a little bit later involved in neutrophil migration, and it makes it a little more confusing, but it's a neutrophil, not a T cell. And this is just kind of going over the same thing again. Invading the pathogen causes cytokine release. And that picture of the macrophage in the tissue, bacteria stimulate cytokines. That regional cytokine release causes vasodilation, upregulates molecules that you would find only on the inflamed vascular endothelium. And the effector T cell has the things that bind to that, binds to the endothelial cell, and then moves through and gets into the tissue. And maybe what's next? It's not working. Okay. Again, I think we've seen this before. That's what's going on the right side of that slide, is the cytokines that were produced have caused not just vasodilation and the leakiness, but the endothelial cells have changed what they're displaying on their surface. Now they have the right kind of molecules to allow the T cells, activated T cells, to recognize them again. Now, I'm going to tell you that, and we're going to see videos of this, the blood flowing through there is fast. And so you're going, well, how in the world can a T cell find that thing and bind? Well, I'm going to show you that. But there's actually that L-selectin that we talked about. There's other selectins that line these places of inflamed vascular endothelium, and cause the leukocytes, like neutrophils, like T cells, activated T cells, to bind temporarily in their role along the vascular endothelium. Red blood cells are flying by, but the white blood cells are rolling along slowly until, boom, they hit something that signals them to stick, and they move through. And so we'll see what that looks like in just a minute. This is kind of what the animation would look like. But this is a neutrophil, and it just says selectin down here. It could be E-selectin on the endothelial cells, because that E is for endothelial. And it's recognizing that same carbohydrate movie that's on the neutrophil this time. It's kind of the opposite this time. And the neutrophil, these two molecules that recognize each other, it's kind of a transient or weak interaction. But the blood is pushing the neutrophil, pushing the neutrophil this way, but the interaction between these two molecules is just tight enough for a little while that it causes the neutrophil, as it's going this way, to kind of roll over. And then this one releases, the next one binds, and it rolls over, and it just basically rolls right down the endothelium. Slowly, so that if it comes in contact with other molecules, like you would find in a very inflamed situation, the neutrophil could bind more tightly and move over. This is what happens when they find those things. So we've got rolling adhesion. Everything you saw on the previous slide is just the first column here. So we've got rolling adhesion taking place. You have additional molecules that then, and one of them here is chemokines. The green dot is a chemokine. There's a chemokine receptor on the neutrophil that recognizes it. And it's hard to see this, but along the top here is some squiggly lines. That's an extracellular matrix where some of these chemokines, they get through and they bind to it. And I don't expect you to know that, but I think you're probably looking at it, how did this green ball just move through there and it's just hanging out there? Well, it's stuck to the extracellular matrix, and it's a chemokine, and it draws these things through. So rolling adhesion, tighter binding, diaphoresis squeezes through, and then it's going to migrate up the gradient towards where those chemokines are being produced, and that's where the infection is. So you get the neutrophils where they need to be.
I have a question.
Yeah.
So going back to that previous slide, so those selectants, are those white blood cells just rolling along all the time? And then if something is wrong, so is that selectin white blood cell interaction normal? What's happening normally?
I don't think so.
Okay.
That's a really good question, because what we do know is that the endothelial cells really increase the expression of E-selectin and also P-selectin on their surface when they become inflamed. And they do it very fast. P-selectin first, because it's actually in a kind of granule, a vesicle, and it moves up right away as soon as they get inflamed. The E-selectin has to be transcribed and put up. But that's a real good question, because you don't going to have the rolling going on all the time. But it's going to be, so if you think about the tight binding, let's go to this next one. Think about the tight binding in the second part here. That's going to be going on, you can think about it as the cytokines to get the selectins up on the endothelial cells, that is extended farther up from the endothelial cells. The tighter binding, like this is showing you ICAM1 and the chemokines, that's going to be closer to where the actual infection is. So the way to answer that is, the rolling's not going on everywhere, but it's going to be extended around the area where there's something has perturbed the endothelium, and you've got the selectins there. And we're going to actually see a picture, see a video of this. Real quick, these are just some things that I want you to be aware of. As far as the phagocytes, what they can do, they have nasty chemicals inside these granules. Some of them, it's just an acidic environment. Superoxide ion, hydrogen peroxide, and some of these other ones that I don't expect you to know all these things. Just I want you to be aware that they assault these pathogens with all kinds of things. And I think we're going to get into infection stimulates macrophages to release pro-inflammatory cytokines. Yeah. Okay. These pro-inflammatory cytokines, which this is just kind of building on what we've already seen, that initiate an inflammatory response. Again, far right, we've got bacteria interacting with the macrophage. The macrophage produces the chemokines that attract the cells, the cytokines that alter the endothelium, and now we've got, in this endothelium, we have some things up on the surface, and it looks like just like black or blue rectangles that would be like the selectins and some of the more tighter binding vascular addressins. And then the leukocytes will go in there, and you get some clotting to kind of get rid of this. So the inflammatory response is going to recruit the effector cells. You're going to have a destruction of the pathogen, blood clotting to help contain the infection, and then repair of the immune tissues. But out of that, you get some pain, you get some fever, you get some redness, you get some swelling from the leakiness and of the endothelium. That's what typically happens, and we're controlling the infection. But on the left side, that's what's happening. This person stabbed themselves with something, got an infection, and we're taking care of the infection, just as we've been talking about. TNF-alpha, IL-1 beta, IL-6. But if you don't contain the infection, say you have more of a systemic infection, then you have the consequences on the right side here. And this is with a gram-negative bacteria, and it would be sepsis, and LPS, or endotoxin, now a systemic. It produces lots of these pro-inflammatory cytokines. And you can see on the right side here, we're going to get systemic edema, decreased blood volume, hyperproteinemia, etc. going on. Bottom line is, too much of this good thing is a bad thing. So if we keep it localized, we take care of the infection, but if we get sepsis, we get the bacteria in the blood, we have too much of these pro-inflammatory cytokines, and it can cause death. We have to do real quick therapy, and one of the things that some of the therapy you might have to do is provide an immunosuppressive or some kind of steroidal type of thing to shut down the immune response, shut down these chemokines. And this is, I just wanted you to be aware of this. We have the different things that these pro-inflammatory cytokines are doing right in the middle hypothalamus. They're gonna tickle the temperature, some kind of temperature signal in the hypothalamus that's gonna cause a more systemic type fever. If it tickles the fat and muscle, then you're gonna have more localized heat being presented. I think we go to, here we go. What I'm gonna do is I'm gonna go to this one first, see if this works. And this is only about 30 seconds long, but it shows you the
Blood vessel endothelium at sites of infection expresses selectin molecules, P-selectin and E-selectin. Leukocytes, such as neutrophils, express the ligand for selectins in the form of the sialylated Lewis X carbohydrate, S-Lex. The binding of the endothelial selectins to S-Lex is weak and cannot hold the cell against the flow of blood. Instead, the cell rolls along the wall of the blood vessel, making and breaking many interactions with the selectins.
next one.
Activated vascular endothelial cells express adhesion molecules, E-selectin and ICAM-1, and display chemokines on the extracellular matrix. Leukocytes initially adhere by binding to selectins, which recognize the sialylated Lewis X carbohydrate ligand, S-Lex. The initial adhesion allows other molecules to bind. LFA1 binds, weakly at first, to ICAM-1. Chemokine signaling converts this weak binding to tight adhesion. The tightly bound leukocyte now migrates across the endothelium, squeezing between adjacent endothelial cells. Chemokine binding now signals the leukocyte to migrate along the chemokine concentration gradient. The site of inflammation is the source of the chemokine gradient. Hence, leukocytes are attracted from the blood towards sites of infection.
I'm going to go back, and now this next one's a little bit longer, but it's actually, it shows you animation, but then it really goes into a mouse. And you're showing this actually taking place in the mouse, and you get a sense of the red blood cells, how fast they're flowing through, and how slow the rolling is taking place. And then we'll take a break.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached